As the country and federal agencies anticipate results from Phase 3 trials for COVID-19 vaccines, public health experts caution that rushing the process could be disastrous. One of the voices arguing for a steady scientific process is Dr. Natalie Dean. In an August 30th tweet, she said premature issuance of an emergency use authorization could jeopardize the conduct of randomized trials and would backfire if the vaccine is not efficacious or if it has safety problems. In her tweet, she continued by saying, critically, we need the process to be transparent and trustworthy. Now, Dr. Dean joins hosts Dr. Eric Topol and Dr. Abraham Verghese on Medicine and the Machine for an in-depth conversation about the goals and the potential pitfalls for COVID-19 vaccines.
Hello, I'm Eric Topol, and with me is Abraham Verghese in a new edition of the Medicine and Machine podcast. And this is a real delight because we've got with us Natalie Dean from University of Florida. And she has a background as a biostatistician, vaccinologist. She worked with the WHO with Ebola and Zika. And now, to me, is a guiding light for the COVID pandemic. So welcome, Natalie.
Hi, thanks so much for having me.
Well, it's a real delight. I've been following your wisdom for all these months of the pandemic, and I'm hoping today we can get a lot of that out for a lot of the people who haven't not on Twitter or not reading your op-eds and articles. And so that's kind of the overall goal. I guess maybe to start, can you give us your overall sense of where we've been since January in the U.S. and where we are today, where we're headed, just a general sense?
Yeah, well, we're struggling a little bit right now. I mean, certainly we had a terrible spring and we were able to turn things around to some degree, but then, we've slipped back up and we're at a weird state with over 1,000 deaths on average per day, and yet people's reactions to it have really changed. And we're heading into this fall, and with schools reopening and colleges reopening and people back at workplaces, and we don't know what's going to happen with the seasonality of the virus. There's just a lot of uncertainty about what's going to happen over the next few months.
Yeah. Well, uncertainty certainly is a theme. Now, you've always kept them more upbeat, like what can we do to make things better? Do you ever lose that sense along the way? Do you ever get a sense of defeatism?
It's difficult to see how things have played out, knowing that there's so much more that we can do. But just like, early on, I listened to another podcast that had on Jim Kim from Partners in Health, and he talked about the early days of the AIDS epidemic where it was exploding in Africa. And, it's just problems can seem really insurmountable, but you just need to get started and do what you can. So even when the problems feel really overwhelming, then whatever little bit you can do to help, that's what I want to do.
Well, that gets us to the positive side is the exit strategy. And let's hear your plan, how we get out of the pandemic for the U.S. What do you think what it's going to take?
It's going to take a very coordinated response that, uses all of the features that we know work. They may not be very exciting. They may not be new, but things like contact tracing, things like, more widespread testing. And certainly I'm really excited about any innovations in rapid testing and more mask wearing, more education and clearer messaging. It's like all these kind of boring public health things that people don't always appreciate as much that they can add up. And there are places in the country that are getting things figured out. Massachusetts and New York, they're figuring out their contact tracing. And it's just, these are systems that didn't really exist before. I mean, they had contact tracing for certain diseases, but not at the scale that we're talking about. And to really get these things to work, it just takes time and trial and error. And But once you have that in place, I mean, we can learn a lot about what are high risk and low risk settings, and we can, have better eyes on the situation and figure out how to keep people safer.
Recently there's been distinguishing contact tracing the forward and backward. Can you explain what that means?
Yeah, so traditionally when we talk about contact tracing, you're imagining that you identify a case and then you track down all of their contacts in order to refer them to quarantine or maybe get them tested so they stay home. So then that prevents future generations of transmission. So that's forward tracing. But that's challenging with this virus because people can infect others before they even have symptoms. So you have to be really fast in order to catch that next generation. Backwards contact tracing, the idea is that you're focusing on trying to find the source of that person's infection. And because we know that this virus can lead to like super spreading events or clusters, if you look backwards, it's possible that by finding the source, there will also be other people infected from the same source. And then if you can find those people and get them tested, then maybe you can identify more chains of transmission and then catch up with those, track down their contacts. So it's a way of looking backwards in order to look forwards and find more chains of transmission. It's also a way to understand the types of sources that appear over and over. Maybe there are certain types of settings like workplace settings or parties or events, things like that, and figure out what are the risky settings.
Abraham, I'm going to get you in here because I know you've got stuff to look at. Question.
Well, we're so pleased to have you on this show, and I'm intrigued by your background working with Zika and Ebola. And I wonder, how that informed you as this new virus came along. Clearly a very different virus, but it seems to me that even with Zika and Ebola, you weren't dealing purely with a virus and a immune response. You were also dealing with all these social imponderables in the countries that were involved. So talk a little bit, if you would, about that challenge.
Yeah, they're so different. The virus is Ebola is just a much more severe pathogen. And so we were able to detect more of the cases. There are, relatively few asymptomatic or very mild infections. Zika was the other end of the spectrum where there were some severe events with the pregnant women and Guillain-Barré syndrome, but then there was a big pool of asymptomatic infections. And here we're somewhere in the middle, but we have, these are very large epidemics. This is a very large epidemic, and it's just affecting so many people. My research has focused on vaccine studies, and so that's primarily I've been thinking about how we make some of these, how we evaluate new vaccines during pandemics and how we make that work. And that has to do with the surveillance systems in place and our understanding of the epidemiology of the virus.
You've been very eloquent about herd immunity, or rather the fallacy of herd immunity. Explain to our listeners, if you would, what that means and the realities of herd immunity for this virus.
Yeah, so I think there's a lot of confusion about herd immunity and what that means exactly. So usually we're used to talking about herd immunity in the context of vaccines. And in that case, it is a safe way to protect people who are unable to receive vaccine. Either they're too young or they're immunocompromised. And so herd immunity, by enough people receiving vaccine, then new outbreaks are not able to sort of take hold, and that protects the people who are not vaccinated. But in this context, we're talking about infection-induced herd immunity. The idea is that people get infected, they develop immunity, and then the idea is maybe that could protect other people from getting infected. But the challenge is that's not a protection strategy, because what happens along the way is all these people get infected and get sick. And so while we may see situations where hard-hit places have accrued some level of immunity and that will impact future dynamics, and whether are we going to see another outbreak in New York City, it's unclear. But what New York City had to go through in order to get to some level of immunity in the population is not something that we would want for other places. And that's why when we talk about what we should do next, we're really focusing on the proactive strategies we can put in place to protect people.
I know that people are often pointing to Sweden as an example of something, but as you point out in some of your Twitter feeds, that's hardly an example of something that we need to emulate, or not for the reasons that they're stating.
Yes, Sweden is a complicated situation. I think it's not always been portrayed accurately in the media. I mean, I think the way that they started is with a lighter touch, and then they have since added things. Then actually now probably what they have in place looks in many ways similar to what other places have. So they started with a lighter touch and have added things, where other places have started, taken more stringent measures and backed off. And I have questions about some of their choices, including they're not using a lot of mask wearing, and but actually, if you look right now at how their numbers are doing, they're not doing poorly. I mean, so it'd be interesting to understand exactly what's going on in Sweden. But I think a big part of it is that they have a very compliant population that is adopting some of the voluntary measures that have been recommended by the government. A lot of people also live alone, spending a lot of time outdoors during the summertime. And so the lessons learned there may not apply to other countries, particularly the U.S., where we don't have the same healthcare system and people may not comply with recommendations.
Yeah, I think that's an important misconception that Sweden just had this let it rip story, when obviously they've had many measures and their numbers are looking good these days. So it's a great point you're making there, Natalie. Now, you talked about the non-pharmacologic interventions, the mask and the distancing and all that. But obviously what is the buzz right now is about this vaccine and the potential of a premature stoppage of a phase 3 trial, or even overriding that we don't have a phase 3 trial. What are your thoughts about where we stand with these 2 U.S. trials that are, I guess, going to have completing enrollment perhaps by the end of September, but obviously not a lot of follow-ups after that point?
Yeah, so, over the last few years, I've been involved with the World Health Organization's Research and Development Blueprint Initiative. So since Ebola, we've been thinking about how do you evaluate vaccines during emergencies, and, how do you make these trials successful, in outbreak and these complex settings. And over, what we've come back to is that you need to make sure you get the evidence in order to inform the decisions. And once you put something in place like an EUA, it's very possible that you may not be able, from that point on, to generate any more evidence. And if you put in place something like an emergency use authorization prematurely, then you can be left in this state of perpetual limbo, where you are not quite sure if the product works. Because if you approve something, but you're not quite sure, for example, the WHO has stated, and the FDA has agreed, that a vaccine should be at least 50% effective, but also we should be able to rule out that the lower bound should not be below 30%. We want to rule out that a vaccine is 20% effective, 15% effective. That would not justify widespread use. And so one concern I have is that these early looks at the data may not provide enough information to rule out that lower bound. I mean, we may see a point estimate above 50%, but are we confident that it's not a low efficacy vaccine? Are we adhering to the guidelines that have been pre-specified for what level of evidence we need? Because if we don't follow those, then we may never be able to go back and figure it out.
Well, you're bringing out that point. Like, for example, the Pfizer CEO has talked about having about 100 events and having a 50% reduction, but the confidence intervals over those point estimates are all over the place. And so to be able to extrapolate that to give a vaccine to tens of millions of people seems like a pretty big reach.
Yeah,
Well,
and there's different.
Go ahead. I'm sorry.
And there's different levels of evidence needed for different situations. I mean, if you're talking about using a vaccine in a very limited way for, like post-exposure prophylaxis, or like a rabies vaccine or anthrax, or, there is different sort of levels of evidence that are needed. But if you're going to be vaccinating millions of people, the level of evidence you need is really high. And I think that's important to reiterate, is that we want to be very confident that our product is safe and effective because we plan to use it in so many people.
Yeah, so different than the vaccine programs that you've worked with. Because now we're talking about billions potentially of people to get a vaccine. So now that also brings up that any given trial will have a limited number of people who are age greater than 65. They won't have children. They're underrepresented of the underrepresented. What are your thoughts about these different groups that are also, we're not going to have sufficient data?
Yeah, completely. That's something that Mark Lipsitch and I have been talking about lately. And we know that these trials, what they're designed to do is to look at the overall trial population. Does the vaccine work or not in this combined population that includes a wide range of ages, so like 18 plus. And but at the end of the day, when we look at the data, we are unlikely to be powered to look at subgroup specific effects. So in particular, we're interested in how well does the vaccine work in older adults, how well does the vaccine work in people who have other comorbidities, and what does that mean for how we plan to strategically use a vaccine? We have, we know that from other vaccines for other diseases that sometimes they don't work as well in older adults. They just don't generate the same immune response. And so, it is possible that we could end up with a vaccine that works well for younger people, but doesn't really work well for older adults. And in that case, then it becomes increasingly important to get, we want more than one vaccine, but we really want more than one vaccine if we have a product that's not working well in older adults. So that really impacts how we think about things like non-inferiority, or these comparisons for because there's so many in the pipeline. We want to find as many as we can that work.
Well, go ahead, Abraham.
I was going to say that there's some estimates that maybe 1/5 of the population might decline the vaccine. And assuming we have an effective vaccine, how does that play into our national strategy? Absolutely.
That's a major challenge. You can have a vaccine that works, but if people are not willing to take it, then that's going to represent a real barrier for achieving anything like herd immunity and for controlling the pandemic. And so that's why, this social and behavioral research is always neglected in these types of pandemics, but it's incredibly important. It's important both for the design and conduct of these trials. These trials have emphasized that they want a broad representation of different minority groups and different, different communities, but they've had challenges enrolling people. And similarly, when we think about, deploying a vaccine and who's going to consent to receive a vaccine, that understanding how people make decisions and what messaging we can use, that is incredibly important. And also just having the process play out in a way that is transparent and trustworthy. And I think even people who would not describe themselves as vaccine skeptics or very pro-vaccine, can see that the process is, that there are concerns about different pressures and people just want to feel confident that the evidence is there, that the product is safe and effective.
Yeah, so Abraham was on the side of the lack of trust and confidence. And I also wanted to get to the people who take the vaccine and think, well, I'm good to go. That is, I'm now immunized, when in fact, that isn't necessarily the case. Can you comment on the fact that this is not sterilization immunity, that people still will potentially get an infection, be carriers of the virus, they will shed the virus, and so that we still are going to be in a phase where masks and all the things that we're doing now will still be useful?
Yeah, absolutely. So we don't exactly know the mechanism by which these vaccines will work. I mean, the ideal is that they can provide sterilizing immunity, but that, given some of the animal data we've seen and given the fact that it's a respiratory virus, that may not be achievable. I mean, it is possible that a vaccine could at least reduce infectiousness. So you may be infected, but you may not be as infectious to others. But we really just don't know. And we also may not really know from the clinical trial data. The clinical trial data is going to tell us that the vaccine prevents disease. I mean, and that is the public health goal, is to reduce morbidity and mortality. So it will tell us that there are fewer people who are symptomatic, but it's possible for a vaccine to make people less symptomatic, but they could still be infectious to others. And so that's another thing that we're going to need more careful study on. Trials include, many trials include infection as a secondary endpoint, and so we'll have some understanding, but there is going to be uncertainty. And also the vaccine is not going to be, it's not like we're going to have 80% coverage right away. It's going to take time to roll out, time to manufacture. And so there's going to be a long period where we need to sort of be full force ahead on all of the other things that we know work. So it's going to, we can't let up on all the other interventions just because, a vaccine has good promising data.
Right. It's vital. Now, to get to the other part of the exit strategy, because we've covered that we're not going to be putting our masks away, and we're also not to have unreasonable short-term expectations of any vaccine, even when it's past phase 3 and shown to have suppression of significant infections. But before that all comes, there's this on the horizon rapid tests. You mentioned them. Can you tell us why, where we stand with that and why that has promise?
Yeah, rapid testing is very promising, and I really hope that it is something that can be approved soon and moved out soon. I mean, we have PCR testing, so the nasal swabs, and those are very sensitive. They can detect even very low levels of virus, but we know that it's a labor-intensive test in terms of actually needs to go back to the lab and someone needs to run something, and there's a process there, and so of course we need to look into continue to look into ways to accelerate that process. But at the end of the day, it's just it's never going to be as widespread as something like a test you could take at home or a rapid test. What rapid tests can do is they give you a quick turnaround and you can use them frequently. And the advantage of that is the goal is to be able to test people even without symptoms so that you can pick people up during their period of high viral load. We know that viral load tends to peak a little bit before people develop symptoms, or sort of around that time. So you may not, you can't just wait until someone develops symptoms to test them. Which is, largely what we've been doing so far. You could imagine testing people all the time, then you could pick people up while they're very infectious to others. And that allows you to then refer those people to isolation, and you can prevent them, prevent those new chains of transmission. So I see it not as a replacement of PCR testing, but a way to sort of expand our reach and be able to pick up people during that period of peak infectiousness.
Stay with us. We'll be right back with Medicine and the Machine.
Welcome to Medscape's latest podcast series, Medicine and the Machine, featuring Medscape's Editor-in-Chief, Dr. Eric Topol, and Master Storyteller and Clinician, Dr. Abraham Verghese. Remember, you can find the latest in medical news, expert perspectives, clinical tools, continuing medical education, and more at medscape.com.
Is there I noticed that when I was reading a bit about your work with Zika and Ebola, that there were different strategies that you use, such as ring vaccination. And I'm wondering, is there room for some sort of innovation in the way we vaccinate, or the way we select people to vaccinate, that might be needed in a situation like this?
Yeah, so ring vaccination, the purpose is to when you identify someone who is sick, you then identify their contacts and their contacts of contacts in which they form a ring around this individual, and you target them for vaccination. This is how smallpox was eradicated in the 1970s. So it vaccinates people who are at highest risk, and it also creates this buffer by which the disease cannot sort of escape beyond that population, that infected person. A challenge here is that most of the vaccines, at least the early ones, are 2-dose vaccines. So they're not as fast to be used in that kind of reactive way, but we can still think about spatial units. So where are the parts of the country where there's most transmission, and how do we want to target those parts of the country? There's been a lot of discussion about vaccine allocation strategies and what is the optimal allocation. Mostly that's focused on sort of individual level features like someone's job or their age group or their presence of comorbidities or their social vulnerability. And so there's been a lot of discussion about allocation strategies. I'd like to see more discussion about the hotspots and how are we targeting hotspots in order to best control the virus.
Now, getting back to the vaccine there, we didn't talk much about safety. What are your concerns regarding a vaccine that is ultimately safe? What do we need to know to be able to make that declaration?
Yeah, so we need, so we need some level of follow-up that occurs shortly after someone receives vaccine. We know that often when vaccines have side effects, they're sort of time limited. They occur shortly after, within a few week period after individual receives vaccine. Like when we've seen Guillain-Barré, something associated with vaccine that's time limited. So we need at least 1 to 2 months of follow-up in enough people after they've received their second dose. The other safety concern that we want to be able to confidently rule out is the possibility for enhanced respiratory disease. And so the discussions I've heard that this would be most likely to manifest, if this were a concern, this would manifest in that when people are infected, they would have more severe disease. So that's sort of gets rolled into how we monitor the clinical trials. And if we saw an excess of hospitalizations among the vaccinated arm, then that would signal that that there was a safety concern with the vaccine. So that goes along with animal data, that goes along with sort of lab data and biological, different biomarkers. And then there's the sort of the longer term safety data. And so we have very less concern that there are going to be long-term side effects. I haven't really heard anyone posit, real long-term side effects that we could expect. I mean, with the dengue vaccine, there was another type of enhanced disease that had to do with once the antibody response started to wane, then individuals were at greater risk of severe disease. So that was something that didn't appear until about 2 to 3 years after those people were vaccinated. But there isn't, really evidence that's a concern. But it's, a challenge is that we're potentially not going to have this longer term, multiple years of safety data before we start using a vaccine in greater amounts. So
well, along with that, there's, I guess, a couple of things. One is we don't know the duration of protection the vaccine might give. Mm-hmm. Yeah. So with respect to the lack of sense of that, I guess that could come from these phase 3 trials. If they keep the arms intact with the placebo group, even if after they have a efficacy established, that's kind of funky, don't you think?
Yeah. So that is that's a question. I mean, I think so once you establish efficacy in an interim analysis, does everyone then receive vaccine? And some places say yes. And I've worked with the World Health Organization's Solidarity Trial, and the plan there is as much as possible to preserve the placebo arm until a vaccine is available, to preserve that time to actually be able to answer questions about long-term protection or long-term safety. So that is an open question. And I think we'll see how that plays out in practice. I think there will be definitely pressure to vaccinate people, but we have to be very clear about what then we're losing in terms of our, the questions that we can answer as a result.
The other thing about safety, just to bring up, and this immunopathologic reaction, apparently in the vaccine trials, only 1% are seropositive when they go in, meaning that we have very few people who've had infection, whereas in the U.S., if you believe maybe 14% of people have been infected by seropositivity. We're not going to be testing that when they get a vaccine. So could we, again, not like the malrepresentative patients we have, people with the underrepresented minorities and the age that I mentioned, but are we going to have a problem where we're going to now put it out to the world with people who've had a prior infection, who get the vaccine, and now we have a somewhat something we don't have our arms around?
Absolutely. Yeah. So we've seen with the dengue vaccine again that there was the vaccine worked differently for people who were baseline seropositive versus people who were baseline seronegative. And, that impacts how these trials are designed. I know the Oxford, I don't want to give bad information, but I believe one trial, maybe the Oxford trial in the UK, excluded people who were baseline seropositive. But in general, I think that what the trials in the US have done is exclude people who were known to have had, were PCR confirmed, they've had confirmed disease. But so maybe that's why the numbers are lower, but there's some of them are still some of them are still seropositive. But yeah, it's basically, we want to make sure that the vaccine is safe for everyone and works well in everyone. And right. So that's why we need to make sure that the population that's within the trial is representative of the population that we're going to use the vaccine in. And so, the questions that we can't answer in the trials, that's where we're going to want to set up post-licensure studies and other types of studies to try and address some of these subgroup specific questions.
Do you think you have enough information if you were offered one of the two vaccines that are being most discussed that you would say, I'll take it?
Right now?
Mm-hmm.
And no, because I would say that if I'm going to receive a vaccine, I want to at least contribute to understanding whether the vaccine works. I mean, so I would participate within the context of a trial. But it's, I think just deploying the vaccine without, generating evidence, that doesn't make sense to me at this point. Yeah. So that would be my main hesitation is are we gathering that evidence that we need to protect the wider population?
That brings me to the WHO in a way, because that's the kind of working together global plan. And you've worked with the WHO, and here the U.S. has pulled out, diverted the funds, and isn't working in COVDEX, which is this coordinated vaccine program. Can you give us your sentiment about this go it alone story that we're seeing?
I think it's unfortunate because the reality is that we are very connected as a global population, and that's how the pandemic was able to spread so quickly. I mean, our world is actually quite small when you see how connected we are, and so a threat anywhere is a threat everywhere. And so if we want to succeed in if we're thinking just about the safety of Americans, then that also means that the world needs to be a safer place. Because we're all mixed up. But of course, also, I work in public health because I'm concerned about people's health everywhere. So, the WHO has a very challenging mission, to work with all these diverse countries who have their own, prerogatives and goals. But they do very important work, which is to bring people together to strategize about, what we should be doing next. And I think they've had, provided a real service, and having sort of clear communications to people and transparency. And, in a time where we haven't heard a lot from the CDC, we haven't heard a lot, about digging through the evidence and what we should be doing. And so the daily press conferences WHO has had have been very valuable. And so, I think it's
they're doing such great things. And I know, for example, they had a meeting with the long COVID folks, at least, a good 60 plus of them, where the leaders of WHO actually sat with them to hear what it was about. And I mean, you don't see anything like that going on here, that's for sure. So I've been admiring their efforts, and it's also, I've gotten some insights through you. The other thing I wanted to get your comments on, we both are on this advisory group for the COVID-19 tracking project, which is a very large group of hundreds of volunteers that are trying to bring together data that otherwise might not be accessible. Can you comment on what that has done for the U.S.?
Yeah, the COVID tracking project has been an incredible asset to all of us, because at a time when we all would have expected the federal government to step up and provide as clean as possible, sort of standardized data set for us to understand what was actually happening out in states, that wasn't available. And so is sort of this vigilante group, a ragtag group of volunteers, came together and started, from the Atlantic, and started collating this data, and it's been a real service, because it's, besides providing the data set, it's provided understanding of the data. It's and it's helped set sort of a benchmark for what we should be expecting from states. It's made states more accountable, because it allows more people to lay eyes on the data, and that's what we need. Is we need, all of us, we need more information, not less, so that we can, make decisions.
Another thing I just want to get at before we wrap up, you've really been extraordinary in terms of recognized clear-eyed thinking, and your posts on social media, and you're just, really become, and by the way, if you aren't following Dr. Dean on Twitter, you're really missing out. I mean, it's worth getting on Twitter just for that. But tell us about how this has influenced your career and your thoughts about, to be able to help provide guidance like this, use social media to try to set things straight or organize your thoughts. What kind of impact has that had?
Well, thank you for your kind words. And it's been a very bizarre time to be, someone who focuses on infectious disease research, and our fairly small-ish community we've gotten a lot more attention. And, just for me, social media was a way. Well, I started off using it really because I was on maternity leave at the time, and I could just sort of tweet while taking care of a baby. And I realized that there are a lot of people who just wanted to learn and understand. And I had this set of experiences from thinking about MERS and thinking about SARS and thinking about, Ebola and Zika and vaccines and all this other stuff, that and ability to translate what other people were doing, and to provide some set of insights. And I realized that there were a lot of people who wanted to learn. And so sort of leveraged my love of teaching. And then it's just, information is moving so quickly, and it's not going through the normal channels. It's coming out in preprints, it's coming out in press releases, and people don't really know how to make sense of things. And I mean, I talk to a lot of reporters. I spend a lot of time talking to reporters, but social media has allowed me to also just kind of talk directly to people. And so I think that's been useful. It's, and it's brought me to a lot of different experiences, chatting with you, chatting with lots of folks. And so one good thing has been that I've been able to, get to know a lot more people in, within my field and in the wider, biomedical field. So that has been nice, because it can be very, it's very isolating to be at home. I think we're all feeling a bit of isolation. So in another way, I've made a lot more connections.
It's been really refreshing to hear from you and to follow your your tweets. I only wish that you were able to influence our national policy more. And if it's not you, I wonder who it is. But clearly, we need this kind of clear thinking and wisdom to guide our vaccine strategy.
Yeah, let me echo with you on that, Abraham. What's also was dismaying to me is here, Natalie's at University of Florida, and Florida is one of the worst hit states in the country, and they didn't even turn to her, as best I know, to help guide them through what was a really tumultuous time. So we're missing out on somebody with just extraordinary wisdom, and hopefully that will change in the future. Natalie, we're so grateful to you to take your time with us today, and we're going to continue to follow you closely because you are the antidote to the infodemic. You are giving us good, straight information, and we're indebted to you for your efforts, really are. Thanks for joining us today.
Yeah, thank you so much. I really appreciate it.
And that's it for this edition of Medicine and the Machine. I am a producer on the show, Nick Andrews. Medicine and the Machine is produced by Medscape editor Robbie Atuma. It's hosted by Dr. Eric Topol and Dr. Abraham Verghese. All Medscape podcasts are produced by the Senior Vice President of Editorial, Dr. Ivan Aransky. Don't forget, there's a transcript of this episode available at medscape.com. You can find a link to that in your show notes. For Medscape, I'm Nick Andrews.